Good day everyone, I'm Bisal Bandari and I'm very excited to present my project on competitive fitness of Curtobacterium flaccumfaciens, Pathova flaccumfaciens lineages. Let's start with agendas. So we'll be first talking about introduction to the problem, and then after that I'll talk about project aims and scope, and then I'll talk about background information. After that I'll provide you my research design, expected outcome, conclusion and references. Let's dive into introduction. The global food security in the face of exponential population growth is a challenge, and agriculture plays a major role in increasing crop productivity and reducing the crop losses. But the major factor here is crop diseases. Crop diseases are causing yield losses up to 20 to 40% worldwide. And in this project, we will be focusing on tan spot because it's a serious disease caused by Curtobacterium CFF, and it has significant yield losses. Mung bean, on the other hand, is the important crop in Australia for domestic and export markets. It's not only important in Australia, it's important all over the world. But in southern and central Queensland, the mung bean growers are increasing. But the tan spot disease is causing significant risk to the mung bean growers because it causes yield losses and compromises market value. In this picture, you can see the symptom of tan spot. It's irregular papillary, tan brown, interveinal necrosis, and chlorotic margin are the symptom of tan spot. In this picture, you can see necrosis, but there is fair amount of chlorosis. And genetic diversity within the CFF poses a risk in mung bean. This is the problem we want to address. There is limited information about different strains of CFF and how they interact with mung bean crop. So investigating competitive fitness of CFF strain will contribute to our understanding of survival, spread and infection severity, providing insight to the disease management strategies. We are assuming that different CFF strains may have different level of pathogenicity. Now let's talk about project aims. So in this project, first aim is to optimize 3 strain-specific PCRSs for various isolates of CFF. We will be using 3 CFF strains, BRIB 70610, 70614 and 70623. After that, we will inoculate. So our plan is to aim is to inoculate mung bean with mixture of each strain and equivalent concentration of pure strain inoculum. After that, we are aiming to compare different disease expression, and then we want to confirm the strain growth dynamics through qPCR when inoculated as pure culture or as mixtures. So this study can help us to develop effective disease management strategy by developing a comprehensive understanding of CFF. So that's the scope of this project. And another one is we will be focusing on multiple strains from the different lineages of CFF. But some of the limitations are like we have limited previous studies on CFF pathogen, which may result in fewer available references and resources for comparison and analysis. We are doing this experiment on control environment in glasshouse, so that may be different from actual field. We have limitation of time. We don't have enough time, maybe we don't have 2-3 years, and we are only using 2 cultivars for this experiment. So maybe the result on other cultivars appear different. So some of the background information for this project are as follows. Competitive fitness in bacteria refers to the ability to outcompete other by efficiently utilizing limited resource in a given environment. And the co-inoculating and single strain inoculation has been done in zebrafish, which is very different from agriculture in mung bean, but some of the information from that study is helpful for our project as well. And in 2019, Begafi and other examined whole genome data from CFF strains linked to the transport of mung bean. This information will be helpful for our experiment, and the project will use unpublished data from CFF genome lineages from CHS. CHS is Center for Crop Health at USQ, and we got some data from there as well. So talking about research originality and motivation, limited knowledge exists on competitive fitness of CFF, and we want to have more information about this. We want to reduce that knowledge gap. That's why we are motivated. And understanding dynamic and interaction of CFF lineages can provide insight to their pathogenicity and spread, and will help to contribute towards the disease management strategies. And we are quantifying CFF after inoculating for the first time, and we are doing this to examine the growth dynamics. So I don't think anyone ever quantified CFF isolates after inoculating, after harvesting and collecting data. So this will be the first time, and this is the originality of our project. So the method of this experiment, this project, will include 17 treatment with different strain combination and controls. We will be using two mung bean cultivars, Opal AU and ZAU. Opal AU is susceptible towards mung bean, towards transport, and ZAU is less susceptible. And we will use 2 replication per treatment. So after that, we will inoculate plants with single and co-inoculation, single strain and combination of strains. After inoculating, so in inoculation, we will use 10 microliter of bacterial suspension, and with the help of acupuncture needle, we will inoculate it into the plants. And after 10 days, we will collect the data. Visual analysis will be done. Sample will be collected, and after collecting sample, they will be dried and biomass will be measured. After that, DNA will be extracted and PCR will be used to quantify the isolates. And after that, with the use of statistical analysis, we will assess the competitive fitness and interaction of CFF pathogen. So if we talk about expected outcomes of this project, we expect that we will have enhanced understanding of competitive fitness of CFF lineages after the completion of project, and we will have knowledge around host responses to mixed inoculation and exploration of growth and colonization differences of isolates in cultivar using PCR and other analysis technique will be more available, and insights into disease management strategies for transport are expected. In conclusion, let's recap this project. So this research problem we are trying to study is genetic diversity within CFF poses a risk in mung bean crop, and we want to explore virulence of CFF strain through competitive fitness study. And our methodology involves optimizing PCR assays, inoculating mung bean, comparing disease expression, and after that confirming strain growth dynamics. And this project will help to provide better knowledge about strain interaction, growth dynamic, and disease management strategies. And it will be helpful in addressing the knowledge gap, improving disease management for sustainable mung bean crop production, and enhancing global food security. Our project findings can be helpful to develop effective disease management strategies in mung bean. So these are the references I took for this project, and I would like to acknowledge University of Southern Queensland for providing me opportunity to do research, and Broadacre Crop Initiative BACI for their the funding body of this project. And I would like to acknowledge my supervisor, course examiner, and all the other people who helped me throughout this project. And this is me doing some field work. Thank you everyone. If you have any question, please let me know. I will be happy to answer them all. Thank you.